Evening, folks. Hope everybody is doing well. Just so you know up front, this is part 2 of my lesson on cell division. If you have not already watched part 1, I recommend you go back and do that first so you have the foundational knowledge you need for this one. Also, if you are currently a student in my class, you should go on Blackboard and get the handout with the lecture notes summary for cell division. That will be quite helpful to have handy for this, both for the diagrams and for the vocabulary that I'm going to give you as we proceed. If you are not a student in my class watching this, because apparently I've got a few non-students watching this as well, I might recommend that you look for some diagrams of some of the terms that I'm talking about on Google as you're watching, just so you have some visuals to go with what I'm discussing here. So we left off last time having identified that there are two fundamental forms of cell division pathways. We've got mitosis, which is used to make identical copies of the mother cell, and we have meiosis, which is used to produce haploid gametes, sex cells, for purposes of sexual reproduction. And I'd like to talk now about those processes, how they work, how they're alike, and how they differ. So let's begin with mitosis. Mitosis is the most common form of cell division. It is engaged in by virtually all cells at some point in their existence. Mitosis is useful for growth, it's useful for repair, and it's useful for some forms of asexual reproduction. Mitosis ultimately is all about making identical copies of your mother cell. You start with a somatic cell, a skin cell, a bone cell, whatever. You duplicate it, you end up with two cells that are just like it at the end of the process. Now, when we're starting out mitosis, we are assuming that our cell has already been through the cell cycle, or most of the cell cycle. It's already been through its G1 phase, in which it did normal stuff till it passed the G1 checkpoint. It has been through its S phase, during which it duplicated its chromosomes, temporarily doubling its chromosome count. And we've been through the G2 phase, the gap2 phase, during which the cell made active preparations for division. Now we get to the beginning of M phase, and it's time to move into mitosis. Remember that starting out, if this is a human cell we're talking about, and let's just assume I'm talking about human cells unless otherwise stated, although the pathways I'm laying out apply to any kind of cell that engages in mitosis, even if the exact chromosome numbers differ from one species to the next. But for humans, remember we started out with a diploid or 2N cell with 46 chromosomes, 23 different ones, 2 of each. But during the S phase of the cell cycle, we doubled that. So now we've got a tetraploid or 4N cell with 4 copies of each chromosome, total of 92 for humans, going into mitosis at the beginning. Now, mitosis has 5 distinct phases. You might have learned 4 phases in high school, or if you've been in school previously, and we'll talk about why in a moment. The 4 phases of mitosis in the order in which they come are prophase, P-R-O-P-H-A-S-E, prometaphase, P-R-O-M-E-T-A-P-H-A-S-E, metaphase, M-E-T-A-P-H-A-S-E, anaphase, A-N-A-P-H-A-S-E, and finally telophase, T-E-L-O-P-H-A-S-E. So they all end in phase, P-H-A-S-E. Now, when you learned about this in high school, there's a very good chance that what they taught you was 4 phases of mitosis: prophase, metaphase, anaphase, telophase. In all probability, you didn't get prometaphase. That's because that's a comparatively recent addition to our understanding of mitosis. We realized that the events that we had been previously lumping together under the heading of prophase did in fact represent 2 separate steps of activity within the cell with different genetic triggers. And as such, it made more sense to break them apart and distinguish them as 2 separate phases in which different things were happening. So what was once called prophase has now been broken into prophase, prometaphase. You'll still see some books calling it all prophase. Some books kind of split the difference and call it early prophase and late prophase. But I want you guys to think prophase, prometaphase, metaphase, anaphase, telophase. Those 5 phases. All right, so we begin with prophase. We're starting out with our tetraploid cell. Remember, it is already prepared for division by duplicating all of its chromosomes, by copying the rest of its organelles and other structures. During prophase, a couple of things are going to happen. First off, I told you about the chromosomes last time and how chromosomes are these packaging systems that condense DNA down very, very tightly. Now, normally, when the cell is not getting ready to divide, the chromosome is kind of relaxed. The chromatid is sort of loose. That makes it easier for the cell to access the genes in the DNA if it's not all tightly packed. But in preparation for division, we want it to be tightly packed to make it easy to move around. So during prophase, the chromosomes are going to become very condensed. They're going to pack down as tightly as they can, squeezing the DNA into as small and portable a unit as possible. A lot of textbooks describing this will say that during prophase, the chromosomes condense and become visible. And the reason we say become visible is because when they're very condensed like that, they're thick enough that you can see them with a conventional light microscope. When they're stretched out, the chromatin is too thin for a standard microscope to visualize. So chromosomes condense and become visible. The other big thing that happens in prophase is that the nuclear envelope begins to break down. Nuclear envelope, remember the membrane surrounding the nucleus, cutting the DNA off from everything in the cell. Nuclear envelope is going to dissolve at this time. It's going to cease to exist as a distinct barrier. That means that the chromosomes are released into the cytosol of the cell. This is the one and only time in a eukaryotic cell's normal activity when the DNA leaves the nucleus, because the nucleus stopped being there. So that is prophase. Chromosomes condense and become visible. Nuclear envelope breaks down. Easy enough, right? So now we move to prometaphase. Now I want you to think back to our lessons on eukaryotic cells. And remember that I told you about an organelle or a pair of organelles called the centrioles. C-E-N-T-R-I-O-L-E-S. Centrioles. You have two centrioles in a cell. Whenever they're not in use, they're normally kept in a single unit called the centrosome. C-E-N-T-R-O-S-O-M-E. Now, during this time, what's going to happen is that we need to get ready to separate the chromosomes. So what's going to happen is that the centrioles are going to migrate to opposite poles of the cell, one on each end of the cell. We talk about cells a lot of times using the same terminology we would use for a globe. It has poles at either end, and it has an equator around the middle. So the centrioles are going to move to the poles at either end of the cell. Then each centriole is going to extend a series of actin microtubules. That is slender tubes made out of the motion protein actin. They're going to reach into the interior of the cell towards the equator. So if you're looking at this as it's happening, it's going to kind of look like you've got this spidery arrangement of little fibers extending out from the centriole at the end and kind of stretching out, curving towards the middle of the cell. Got this happening at both ends. So you end up with this kind of football or almond-shaped arrangement of fibers reaching towards the cell's equator. That structure is called the mitotic spindle, M-I-T-O-T-I-C, spindle, S-P-I-N-D-L-E. Sometimes it's called the spindle apparatus. You can use either term. I'm fine with either one. The spindle apparatus is going to be the structure that actually separates and moves the chromosomes during this process. The individual little microtubules within it, you can just call them microtubules. You can call them asters. That's an older name for them, A-S-T-E-R. You don't hear that one much anymore. Or you can call them spindle fibers or spindle threads. You'll hear all those terms used for them. I don't care which one you use. I usually say spindle fibers. All right. But we form these actin spindle fibers reaching into the interior of the cell. Last thing that's going to happen in prometaphase is that, I think I mentioned to you all last time, that as parts of the centriole, not centromeres, the centromeres. The centromere is not part of the centriole. Don't get those two terms confused. I've got a lot of centro things going on here. Centriole is the organelle making the spindle apparatus. Centrosome is where the centrioles live when we're not using them. Centromere is the little spherical mass of proteins holding the two sides of the chromosome together. You might recall, I think I said this in the last lesson, and if I didn't, I'll say it right now, that part of the centromeres is going to be a group of proteins called kinetochores. 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 The kinetochores are vital to the process of moving the chromosomes around during mitosis. And at the end of anaphase, the spindle fibers are going to attach to the kinetochores, getting ready to move those chromosomes around. So, in quick summary, during prometaphase, centrioles migrated to either pole of the cell. They stretched out their actin microtubules to form the mitotic spindle, and then those spindle fibers attached to the kinetochore proteins that are produced by the centromeres. Sound good? Okay, so that's prometaphase. Now we move to metaphase, where things start to get interesting. During metaphase, the spindle fibers are going to poke and nudge the chromosomes and line them up along the cellular equator. We're going to get a line of chromosomes forming along the cellular equator, which we often call the metaphase plate. Now remember, we have sister chromatids here, right? We talked about that last time, that all the chromosomes got duplicated. The duplicates are called sister chromatids. Remember, they're still joined to each other by the centromere. So you have kind of these X-shaped or cross-shaped chromatids that are all joined by one centromere, 4 chromatids, 4 arms, all reaching towards that one central point. And you got 46 of these. Each sister chromatid is attached to its original, total of 92. Lined up down the cellular equator. So one of the sister chromatids is oriented towards the north pole of the cell, and one of the sister chromatids towards the south pole. So 46 chromosomes oriented north, 46 oriented south. A full set, in other words, pointing in each direction. Once the spindle fibers have nudged the chromosomes into that position, we've got this line of chromosomes along the equator of the cell. That line is sometimes called the metaphase plate. So we just formed the metaphase plate. Now that we got that metaphase plate, the spindle fibers are going to attach directly to the centromeres of the chromosomes. We're going to have one spindle fiber on each side, joining those centromeres from the north pole and the south pole simultaneously. So in other words, every one of those 46 pairs of sister chromatids is going to have 2 spindle fibers attached to it. Or in other words, every one of the individual 92 chromosomes has 1 spindle fiber attached to it. 46 from the north pole of the cell, 46 from the south. So that is metaphase. In quick summary, during metaphase, the spindle fibers moved the chromosomes into a line along the cellular equator, forming the metaphase plates. Once they were lined up, the spindle fibers attached to their centromeres. All right, are we good? We good? We're good. Okay, we move to the next one, anaphase. For reasons I am not clear on, this is one of the 2 most misspelled words in the entire class. People want to make it A-N-E or A-N-O or A-N-I, anything but A-N-A. It is A-N-A, anaphase. During anaphase, what's going to happen is the spindle fibers are going to shorten. They're going to begin retracting back towards the centrioles. You might think of it like a fishing line that the centriole is reeling in. Now, as the spindle fibers shorten, they're going to pull chromosomes along with them. The centromere is going to split. The sister chromatids are going to separate from each other, and they're going to move towards opposite poles of the cell. So at the end of anaphase, what we're going to have is 2 separate but equal groups of chromosomes, genetically identical groups. Each one is a complete set of chromosomes, 46 near the north pole of the cell, 46 by the south pole of the cell. We've successfully divided our chromosomes now into 2 distinct groups. And that, folks, is anaphase. In summary, spindle fiber shortened, chromosomes were pulled towards opposite poles of the cell, sister chromatids separated from each other. All right, you good? All right. So we bring to the last phase of mitosis, which is telophase. This is, by the way, the other most misspelled word in the entire course for some reason. It is T-E-L-O, telo, not T-E-L-E, not T-E-L-A, telophase. During telophase, we're basically going to reverse a lot of what happened in prophase and prometaphase. Spindle apparatus is going to dissolve. The spindle fibers are going to break down, leaving the chromosomes at the pole of the cell. Centrioles are going to move away from the poles of the cell, return to the centrosome. Their job is done. Then we are going to rebuild the nuclear envelope around each separate group of chromosomes. We're going to encase each of those chromosome sets in its own nuclear envelope. So at the end of telophase, what we're going to have now is a cell with two nuclei, one in each pole. Each of those nuclei contains a complete diploid set of chromosomes, 46 chromosomes in humans, 23 pairs. All right, that is telophase. In summary, spindle apparatus broke down, centrioles moved away from the poles, nuclear envelope is rebuilt around each separate group of chromosomes. All right. And you know what, folks? That was mitosis. It's the whole process. And you can see that basically what we just did here is we took all those chromosomes that we had duplicated during the S phase of the cell cycle, and we sorted them into two equal groups. We gave this cell two separate nuclei so that each of the new daughter cells we are going to form has a nucleus with all the chromosomes that it needs. That is what mitosis is for, folks. It's for separating the chromosomes into two equal groups. But we're not quite done yet. We finished mitosis. We have not finished M phase. The M phase of the cell cycle has one other event that's going to happen, and that is cytokinesis. C-Y-T-O-K-I-N-E-S-I-S. Cytokinesis is going to be the process by which the cell divides up its cytoplasm and other contents into two new cells. What's going to happen during cytokinesis? We've got this binucleated cell that we formed during telophase, this cell with two nuclei, one in each pole. What's going to happen is that cell, we've got this whole cell here, right? It's going to begin to pinch inward. You can get an indentation forming down the middle of the cell. The membrane is beginning to fold in on itself. Whenever anything folds inward on itself, we say that it is invaginating. I-N-V-A-G-I-N-A-T-I-O-N, invagination. So the membrane, got this whole spherical kind of membrane, right? Let's pretend this is a sphere. And it's beginning to pinch inward from both sides. It's invaginating. It's folding in on itself. And as it does, the cell is stretching more and more into two distinct compartments, pulling more and more away from each other. We're getting this groove forming down the middle. You can see where that barrier of membrane is getting bigger and bigger between them, and the indentation is getting deeper and deeper. We call that groove the cleavage furrow. Cleavage, C-L-E-A-V-A-G-E. Furrow, F-U-R-R-O-W. Cleavage means to separate. To cleave is to cut things apart. A furrow is a groove or a channel. So we are forming this cleavage furrow. It's getting deeper and deeper and deeper. The two halves are stretching more and more apart from each other. Finally, the pinched inward part meets in the middle. The cleavage furrow meets in the middle. We now have two separate compartments, which separate from one another in a process we call cleavage. And at that point, folks, we now have two distinct daughter cells, each one its own independent entity, each one having received half of the cytoplasm and the organelles and other structures involved in the original cell, and each of them having received one of those two nuclei. We have just built two brand new diploid cells, each of which is a copy of the original mother cell that we started with. 2 identical daughter cells. All right, so that, folks, is mitosis followed by cytokinesis. Now that we're done with that, M phase is over, and each of those new daughter cells goes into its own G1. It begins resuming its own activities. Cell cycle starts over. All right, sound good? Now, to finish out mitosis, just to kind of close this off, I wanted to point out that there are some kinds of cells that don't do this. There are a few kinds of cells that are said to be non-mitotic. A non-mitotic cell is a cell that does not divide. Now, the most common reason for a cell to be non-mitotic is that that cell started off as a different kind of cell. It started off as a stem cell of some sort. You've all heard the term stem cells, I'm sure. Stem cells are embryonic cells that form during your development. The cool thing about stem cells is that they can turn into other kinds of cells. Now, when you first exist, when you're just a ball of cells that hasn't gotten any special features of any kind yet, you're just a ball of stem cells. And eventually, all of those stem cells are going to go through a process called differentiation. D-I-F-F-E-R-E-N-T-I-A-T-I-O-N. Differentiation. When a stem cell differentiates, it transforms into another kind of cell. It assumes a specific cellular identity. Now, some stem cells remain into your adult body. You right now sitting here have stem cells in your body that have not differentiated. Sometimes those stem cells will undergo mitosis and replenish themselves, because sometimes we have to keep on making certain kinds of cells over and over again. Your body always needs new red blood cells and white blood cells. Your body always needs new skin cells. So the stem cells that give rise to those things have to be continuously dividing, undergoing mitosis to keep new ones forming. But some types of cells, once they have differentiated, do not divide again. Once they've assumed a specific cellular identity, they cease division altogether. They're basically in G0 for the remainder of their existence. Those cells are non-mitotic. Only way to get more cells of that kind is through the differentiation of more stem cells. A couple of examples of non-mitotic cells that are probably known to you. Most well-known instance, classic example of non-mitotic cell would be neurons. N-E-U-R-O-N, neurons. If you do not know this already, neurons are the communicating cells of your nervous system. They are the cells of your nerves, your spinal cord, and your brain that actually send signals back and forth and communicate with one another to transmit information. Neurons form from stem cells, but neurons themselves are non-mitotic. They do not have centrioles, and as a result, they are not able to divide. This has important health consequences for you because if you have nerve damage, if a nerve is cut, your body generally has a very limited ability, if any ability, to heal that damage because we cannot grow new neurons. If the other nervous cells around the neurons, which are called neuroglia, are damaged, they can regenerate. So minor nerve damage can sometimes be repaired. But if the neurons themselves are damaged, it's game over. That nerve will never function properly again. This is why if you get your spinal cord cut, you lose the ability to walk. This is why stroke patients who have suffered brain damage often never gain back their full faculties. I got a little patch of skin right here on the inside of my left ring finger, right about there at the tip. You poke that with a needle, it won't hurt me at all. When I was in high school, I was hanging up bunting for a graduation party. I was using a staple gun, put my hand too close to the staple gun, staple right through my finger. Apparently I cut a nerve, and now I've got a little dead patch on the side of my fingertip that 20 years later, I still can't feel. So, we're, by the way, incidentally really interested in stem cell therapies for that exact reason. There's a lot of research being done with stem cells to see if we can find ways to trick stem cells into growing into specific other kinds of cells that we desire and using them to repair nerve damage in that way. Right now, there's an experiment going on at Emory University in Georgia involving stem cell injections into the spinal cords of paralyzed soldiers to try to find out if we can make them walk again. And we don't know how that's going to work yet. It's a long-term experiment. The identities of the soldiers involved are being kept under wraps while they're doing it for privacy reasons, but my contacts at Emory tell me it's looking pretty good. So I think that we may be able to find a medical way around this problem sooner rather than later with the benefit of stem cells. Other kinds of cells that are non-mitotic, and this is not a comprehensive list, but a few other ones that are especially important, erythrocytes. E-R-Y-T-H-R-O-C-Y-T-E. Erythrocytes. The prefix eryth means red. Cyte is cell. Erythrocytes are your red blood cells. You've probably all heard of these. Red blood cells are the ones that carry oxygen in our bodies. They deliver oxygen to your other tissues. They carry away carbon dioxide waste. Now, red blood cells are designed to maximize oxygen carrying capability. And that means that almost everything in them that is not involved in oxygen carriage has been removed. Red blood cells do not have organelles. They do not have a nucleus. They don't even have mitochondria. They're the only cells in your body that are fully anaerobic because all of that space has been taken up with the protein hemoglobin, which is the oxygen-carrying protein that I told you all about way back when we discussed proteins. So a red blood cell is more or less a sack of hemoglobin. And because it doesn't have any organelles, including no chromosomes to separate and no centrioles to separate them, red blood cells do not divide. They are non-mitotic. A red blood cell only lives for about 120 days and wears out. Your body has to continually replenish them. That's what your bone marrow is for. Bone marrow is full of stem cells called megakaryocytes, and those stem cells continuously divide, not only to make more megakaryocytes, and that's megakaryocyte, but megakaryocytes not only divide to make more megakaryocytes, but also to make both red blood cells and white blood cells, which are the agents of your immune system, which also have to be made pretty swiftly in response to infection. And finally, last one I wanted to mention, cardiocytes, sometimes called cardiomyocytes, C-A-R-D-I-O-M-Y-O-C-Y-T-E. Cardio for heart, myo for muscle. Cardiomyocytes are the cells of the heart muscle. You know that your heart is a muscular organ. It has a special kind of muscular tissue in it called cardiac muscle, which is only found in the heart. And the cardiocytes that make it up are non-mitotic. The only way that you get them is through the division of stem cells found in the tissue of the heart called satellite cells. When you have a heart attack, portions of your cardiomyocytes die. And those dead cells are replaced by scar tissue, permanently damaging that portion of the heart because you cannot divide cardiocytes to make new ones. We have learned that there are a limited number of stem cells in the adult heart, which can differentiate to form some new cardiomyocytes from time to time, but not enough to repair significant damage. So the good thing is to try not to have a heart attack in the first place. These are just some examples of non-mitotic cells. Not a comprehensive list, but gives you an idea about some of the ones that do not divide and why, and the consequences of that for us. All right, so that's mitosis. Mitosis is great if what I want is identical copies of my mother cell. But what if that's not what I want? What if somatic cells, normal body cells, are not what I'm interested in? What if I want gametes, eggs and sperm, sex cells for reproduction? That's when we need meiosis. Meiosis is not about making identical copies of the mother cell. It is about making haploid or 1N gametes. At the end of meiosis, we're going to have a grand total of 4 haploid gametes from that original diploid mother cell. Those gametes each only have 23 chromosomes, 1 copy of each. The whole purpose of them is to fuse with another gamete, egg and sperm, in order to make a new individual. Now, I said a few things about meiosis in the last lecture, and I want you to keep them in mind as we go along here. For starters, meiosis only happens to a very specific population of cells. It only occurs in sexually reproducing organisms. It only occurs in the reproductive organs called the gonads, and it only happens to a specific kind of cell in the gonads known as germline cells or germ cells. Those are the only cells that ever engage in meiosis. In females, they typically start engaging in meiosis and producing gametes before the female organism is even born. In males, that typically do not begin doing that until puberty. They can also undergo mitosis. That's how you get more germline cells. But they're the only cells that ever experience meiosis. Eggs and sperm themselves do not experience meiosis. They are the product of meiosis. They do not go through it. Indeed, eggs and sperm in general are non-mitotic. Once they're produced, their job is to be fertilized. That's all they do. They don't make more of themselves. So, how's this going to work? Meiosis, remember, proceeds in 2 rounds of division, not one like mitosis. So we're going to go through a 2-cycle process of meiosis. We refer to the 2 parts as meiosis 1 and meiosis 2. Roman numeral 1, Roman numeral 2 to designate them. Now, meiosis evolved from mitosis, and a lot of what you're going to hear about here is very similar. A lot of it is no different than what happened in mitosis, but there are going to be some crucial tweaks along the way to make the process come out differently. In fact, the phases of meiosis have the same names as the phases of mitosis: prophase, prometaphase, metaphase, anaphase, telophase. Same 5 phases. To distinguish meiosis 1 from meiosis 2, we will put a Roman numeral 1 or 2 after the phase name. So we might talk about, say, metaphase 1 or anaphase 2. And when you're talking about meiosis, make sure you distinguish them so we know that's what you're discussing. All right, so meiosis begins more or less the same way that mitosis did. We start off with a diploid mother cell, in this case, a germline cell found in the gonads. That cell has already been through its whole interphase. Remember, interphase is all the stuff prior to M phase, G1, S, and G2 phases. So it's been through its normal activity up till it committed to division in G1 at the G1 checkpoint. It has duplicated its chromosomes during the S phase, doubling its chromosome counts, and it's been through the process of preparation to divide in the G2 phase and past a G2 checkpoint. So what we're starting with now is a tetraploid mother cell with 4 copies of each chromosome, total of 92 chromosomes in humans. That's what goes into meiosis 1. All right, so prophase 1. Prophase 1 is the longest part of meiosis. 90% of meiosis is spent in prophase 1, and it is the part of meiosis that is most different from mitosis. Some of the most important things that happen to make meiosis distinct happen here. Now, prophase 1 starts off looking an awful lot like prophase of mitosis. Chromosomes condense and become visible. The nuclear envelope dissolves, releasing the chromosomes into the cytosol. Okay, so far sounds pretty familiar. But here's where things start to get interesting. During prophase 1, now, thing that you need to know, you normally, in all your somatic cells of your body, including germline cells, you normally have 2 copies of each chromosome, right? Chromosomes are paired. You have a maternal homologue, that is the copy of that chromosome you got from mom, and a paternal homologue, the copy you got from dad. So if I look for chromosome 2, I'll find 2 copies of chromosome 2, one maternal, one paternal. I can do a karyotype on you and your parents and match up and say, yep, that one right there is the paternal one, that one's the maternal one. I can see where those genes match that parent. Well, what I didn't tell you before is that under normal circumstances, the maternal and paternal homologues do not hang out together. There's nothing saying they have to stay near each other in the nucleus. They're going to just be wherever they happen to be until prophase 1. In prophase 1, the maternal and paternal homologues are going to find each other. They're going to pair off. So in other words, your copies of chromosome 1 are going to pair off with each other. Your copies of chromosome 2 are going to pair off with each other, and so forth. Once they have done that, the chromatids, remember the arms of the chromosome, are going to overlap each other. They're going to overlap, they're going to cross over each other like this, and they're going to fuse together at the point of contact. So let me just draw you a real quick little sketch here to show you what this looks like. So something like that, right? Now, in the diagram on the right, I only drew one set of sister chromatids in each pair because that would have gotten really messy if I was trying to draw that in 3 dimensions and show how both sets of sister chromatids were doing that. But you can see where the chromatids of the maternal and paternal chromosomes are now overlapping each other. They've joined together at the points of contact, those points where they're meeting right there, and there. Those points of contact that form between them are called chiasmata. C-H-I-A-S-M-A-T-A. Chiasmata. If you're talking about just one of them by itself, the singular form is chiasma. C-H-I-A-S-M-A. Chiasma. So, the maternal and paternal homologs have met up with each other. By the way, the process of them pairing off, when maternal and paternal homologs pair off with each other, that is called syngamy. You can see that word there, syngamy, S-Y-N-G-A-M-Y. That's when the maternal and paternal pair up. Then they're going to overlap their chromatids, and they're going to join together, forming chiasmata. Once they're joined in that way, they're going to stay joined together until anaphase 1. They're going to be paired off for a while. Now, here's the coolest part. After they have joined together in this way, these two chromosomes are going to swap some of their genes back and forth at the point of contact where they joined with each other. Those chromosomes are going to switch some of their genes. In other words, some of the genes that started in the maternal copy of that chromosome are going to swap into the paternal copy. And likewise, their paternal equivalents are going to swap into the maternal copy. In other words, right now in your body, you've got somatic cells full of chromosomes. If I look at them, I can look in each of those cells and say, that's the chromosome that came from your mama, that's the chromosome that came from your daddy. Those are just pure chromosomes, maternal, paternal. But crossing over happens, and they're not pure maternal and paternal anymore. Now they're going to exchange some genes from each other at that point of contact. And now this one is mostly maternal, but with a little patch of paternal genes in there. This one is mostly paternal with a little patch of maternal genes. They have now mixed up the genetic information that you inherited from your mom and your dad. They're no longer purely maternal and paternal, but a little of each. Cool thing about this, it is completely random where the chiasmata form. You never can predict ahead of time which spot they're going to join up with each other. It is random which genes and how many genes switch each time. In other words, sometimes you're going to swap a bunch, sometimes you're not going to swap many. And it can be at any point along the arms of the chromosomes. So this is different every time. Every single time you engage in meiosis, it's going to be a different combination of genetic information forming. A different combination of now chromosomes that are mixed maternal and paternal material. Now remember, your gametes are going to be haploid. They're going to have one chromosome out of each pair, which means that when these go apart from each other, the gametes we are going to get are not going to be genetically identical. And this is why, because of crossing over, mixing up the genetic information. Every egg or sperm you produce is going to contain half of your DNA, 50%. But thanks to crossing over, it is a different mix of genes each time. In other words, sorry, my camera did something weird for a second there. In other words, folks, you are definitely half each of your parents, but you're not guaranteed to be 1/4 each of your grandparents, because maybe the sperm that made you happened, by luck of the draw on that occasion, to have a few more genes that came from your grandpa than from your grandma. Maybe the egg that made you had a few more genes from your maternal grandmother than from your maternal grandfather. So you might be like 30% one grandparent on one side and 20% the other. There's no guarantee that it's going to be an equal sharing because, again, we can't predict which genes are going to cross over and how many genes are going to cross over. It's different every time. And this, folks, is why every sperm or egg is genetically unique, and therefore why every child from the same set of parents is genetically unique. Because even with those same parents involved, they're mixing up the genes in different ways. On average, you share about 50% of your DNA with a blood sibling with the same set of parents as you. All right, so that's what we did during prophase 1. We did the breakdown of the nuclear envelope, chromosomes condensed and became visible. The chromatids, syngamy happened where the maternal and paternal homologs found each other and paired off. The chromatids overlapped and formed chiasmata. And then finally, we swapped some genes back and forth between them. By the way, that swapping process has a name. It is called recombination. More commonly, however, biologists just refer to it as crossing over. So recombination or crossing over is the switching of genes at the chiasmata that happens during prophase 1. So that's prophase 1. Chromosomes are out of the nuclear envelope, condensed, paired off with their maternal and paternal homologs, formed chiasmata, crossing over took place, mixing up their genes. All right, sound good? Prophase 1, most complicated part of meiosis. It gets easier from here. Prometaphase 1. Honestly, I have nothing special to say to you about prometaphase 1 because prometaphase 1 is just like prometaphase in mitosis. No differences. Centrioles migrate to the poles. We form a spindle apparatus. Spindle fibers attach to the kinetochore proteins. Everything that happened in prometaphase of mitosis happens in prometaphase 1 of meiosis. Easy, right? We move on to metaphase 1. Metaphase 1 is mostly like metaphase of mitosis. Spindle fibers are going to nudge the chromosomes into a line down the cellular equator, forming the metaphase plates, and then they're going to attach to their centromeres, one on each side. Now, one difference. Normally, whenever you do this, when this happens in your mitosis, you're going to find that you've got 46 identical chromosomes going north, 46 identical chromosomes going south, one paternal and one maternal out of each set. However, when you line them up on the metaphase plates for meiosis, the maternal-paternal alignment of each chromosome pair is random. In other words, there is no guarantee that you are going to get two copies of you're going to get one maternal and one paternal chromosome out of each pair going towards each pole of the cell. Maybe in one pair, you're going to get two copies of the maternal, and in another one maternal and one paternal, and the next one, maybe both paternal. It's random for each chromosome set. This further mixes up the genetic information. It's another way that we make genetic uniqueness in the offspring. That's really the only difference between metaphase 1 of meiosis and metaphase of mitosis is the random alignment of maternal versus paternal chromosomes on the metaphase plate. You're still going to have 46 chromosomes going north, 46 going south. Still going to be, pairs, two copies of chromosome 1, 2 copies of chromosome 2. There's just no guarantee that it's going to be a maternal and a paternal homolog of each. All right, we good? Okay, anaphase 1. Anaphase 1 looks mostly like anaphase of mitosis. Spindle fibers shorten. Chromosomes are pulled towards opposite poles. Sister chromatids are separated. One difference that we see here has to do with the chiasmata. I didn't say this before. I should have said this in the last step. But during metaphase 1, the chiasmata stretch out so that the chromatids are only touching at their very tippy ends. During anaphase 1, the chiasmata finally break, and the chromatids that have been in contact with each other since prophase 1 are now separate. That's really the only difference between anaphase 1 of meiosis and anaphase of mitosis. Either way, you've got 46 chromosomes moving north, 46 moving south. You're going to end up with one complete chromosome set at each pole of the cell. And the chiasmata broke along the way. Had to separate them. All right, and that brings us at last to telophase 1. Telophase 1, Nothing special to say here, folks. It is just like telophase of mitosis. Spindle fibers break down. Centrioles move away from the poles. We rebuild the nuclear envelope around each separate group of chromosomes. So at the end of telophase 1, what we now have is a cell with 2 nuclei, each with a set of 46 chromosomes, 23 different ones in pairs, but thanks to crossing over and thanks to that further admixture during metaphase 1, they are not identical. They are not genetically identical nuclei. Each one is going to contain a slightly different mixture of the genes you got from your mother and the genes you got from your father. So that, folks, is meiosis 1. And meiosis 1 is where the real action happens. This is where we really bake in that genetic distinction that is so characteristic of sexually reproducing organisms. Now, following meiosis 1, in most species, cytokinesis will happen immediately. And we will separate that mother cell into 2 diploid daughter cells, each of which has 46 chromosomes, but again, which are not genetically identical to each other. Now, in some species, mostly in insects, there's no cytokinesis till the end of meiosis 2. That's unusual, however. The norm is to undergo meiosis at the end of meiosis 1, and for the sake of our discussion here, we're going to assume that's what happened. That's what happens in us. Now, following cytokinesis, when the M phase is done, the cell is going to go, each of those 2 new daughter cells we formed, those non-identical daughter cells that came out of meiosis 1, each of them is going to go through a shortened interphase. Each one will go through a G1 phase, featuring a G1 checkpoint, and then a G2 phase, featuring a G2 checkpoint, before they move on to meiosis 2. You might have noticed no S phase in there. In other words, there is no duplication of chromosomes between meiosis 1 and meiosis 2. And that's super important, folks, because what that means is that we start meiosis 2 with not a tetraploid cell, but with a diploid cell. Each of those 2 diploid cells that we made during meiosis 1 is going to go on separately to meiosis 2 without duplicating its chromosomes in between. So that is our shortened interphase. And now we move into meiosis 2. Each of our 2 cells that we just made goes through it separately. I actually don't have much to say about meiosis 2, folks, because meiosis 2 is pretty much the same as mitosis. Most of the events that happen in mitosis happen exactly the same in meiosis 2. Prophase 2, chromosomes condense, nuclear envelope breaks down. There's no crossing over in prophase 2, there are no chiasmata, there is no syngamy. That stuff only happens in prophase 1. Prometaphase 2, just like prometaphase as you've heard about it the whole time. Centrioles move to the poles, we form the spindle apparatus, we attach the spindle fibers to the kinetochores. Metaphase 2, light up the chromosomes along the cellular equator, form the metaphase plates, spindle fibers attach to their centromeres. Here's the first time something's really different. We only lined up 46 chromosomes along that equator this time, not 92, which means that with half of them being attached to one spindle apparatus and half to the other one, we now have only 23 chromosomes oriented north and 23 oriented south. Anaphase 2, pretty much the same as what you've been hearing. Anaphase 2, you'll have spindle fibers shorten, chromosomes are pulled towards opposite poles of the cell. There's no breaking up of sister chromatids here because that happened in a previous stage. These are just separate chromosomes now. At the end of anaphase 2, I will have 23 chromosomes at the north pole of the cell and 23 at the south. Telophase 2, just like telophase as you've been hearing about it. Nuclear envelope reforms around the chromosomes, spindle apparatus breaks down, centrioles get out of the way. And so at the end of telophase 2, we now have a cell with 2 nuclei, one on each pole, but each of those 2 nuclei only has 23 chromosomes in it, unpaired chromosomes, not 46. And thanks to all the scrambling and mixing up that we had during meiosis 1, those groups of 23 chromosomes are each genetically distinct. Each is half of your genetic information, but it's never the same combination of genes choice. Following telophase 2, we will always undergo cytokinesis. The cells will do everything I talked about before. We'll form a cleavage furrow, will invaginate, separate out the two, cleavage happens. And now we have two daughter cells. But we did this twice, remember, because we had two daughter cells at the end of meiosis 1. Each of them went through meiosis 2. So at the end of meiosis 2, we have a grand total of 4 daughter cells, 4 haploid or 1N daughter cells, each of which has a single copy of each chromosome. And now we have just formed 4 haploid gametes, sex cells that are ready to go on for reproductive purposes. Now, I should point out here, they're not entirely done at that point. There are a few other things involved in making an egg ready to be fertilized, making a sperm ready to fertilize. But the fundamental separation of chromosomes to give each of them 1/2 of a genetic set is finished. And that, folks, is meiosis. I have run slightly past my usual runtime of about 50 minutes, so I think this is a good time to call this short. In our next lesson, we're going to talk about fusion of egg and sperm a little bit. We're going to talk about what happens when you have the wrong number of chromosomes, and we're going to talk about how a thing called twinning happens. Sounds good? All right. Hope you're all having a good evening. I'll talk to you later. Good night.